Welcome to the Faculty Focus podcast, live from our annual Faculty Research Day with your hosts
Marleen
and
Teun,
highlighting the research of our professors and PhD researchers.
Hi again. Next on the podcast we have Emile VErhulst. Welcome, Emile.
Thank you.
Your topic of today is From Target to Therapy, FAP inhibitors and their potential in cancer theranostics.
That's right.
Can you briefly introduce yourself to our listeners?
So my name is Emile. I did both my bachelor's and my master's at the University of Antwerp.
In pharmacy?
Yeah, so my bachelor's in
Pharmaceutical
pharmaceutical sciences to be correct, indeed, and then my master's in drug development. But I chose to do a minor in biopharmaceutical sciences because I already knew that I didn't want to do something in the pharmacy, but definitely something in industry or in academia. And also in my master's, I got to know a PhD, which seemed nice to me to do because you could specialize in a certain area, and that's why I ended up doing a PhD currently in the medical biochemistry lab. Yeah.
Perfect. First, I would like to come back to your title and address the elephant in the room, or rather the dinosaur in the title of your presentation.
Yeah.
What type of animal is a theranostics?
So theranostics actually is a combination of therapeutics and diagnostics. So currently a lot of possible drugs are being evaluated in clinical trials towards FAP, but they're all to be used as potential diagnostics. And what we want to do is try to convert or mimic these compounds, but then to be used as therapeutics and not only as diagnostics. But there is one major issue, is that these compounds, they don't stay long enough into the tumor. And we want to try and modify the chemical structure of these compounds to be able to use them as therapeutics. Yeah.
So that they stay also longer in the tumor.
Yeah, and that's
and have
necessary.
a bigger effect.
Yeah, true. Because currently they don't stay long enough, and the effect is not enough to cure the tumor. Yeah.
I've seen some nice pictures of you in a cycling outfit. Well, imagine
That's
that
right.
you ran into an old friend from your cycling days and you have to give them a scoop about your research. And how would you explain your research so that they understand?
So my focus of my research is a very specific protein, as you said already, FAP, or incomplete fibroblast activation protein alpha, which already tells in the name actually. It's only expressed on activated fibroblasts. And what is unique in a tumor is that we have permanently activated fibroblasts.
And those are types of cells.
Yeah,
Right.
indeed. And they express, so that my protein is present on these cells.
Okay.
And actually only on these cells and almost anywhere in the body.
Okay. So that makes it very interesting.
That makes it
to
very
study them.
interesting, and that's why it's so a hot topic today, actually. Yeah.
Okay. And you sometimes put an I behind the FAP.
Yeah, that's
what is
true.
the I for?
So, yeah, FAP stands for the protein, but the I stands for inhibitor. And that's actually, the compound that we make. It's an inhibitor.
Oh, okay.
So we want to inhibit or put a brake onto the FAP, the enzyme. Yeah.
Okay, and how difficult is it to make a perfect theranostic FAPI? Can you say FAPI?
Yeah.
Okay.
FAPI, FAPI, it's the same, I say. So it's a collaboration between our lab and then the medicinal chemistry lab. So they mostly do the synthesizing, so the making of the compounds.
Okay.
And we do the actual screening of the compounds, and we then communicate and tell which compounds have the most potential to further go into possible clinical trials.
Okay.
Yeah.
What are the pitfalls of problems you encounter when making such a FAPI?
So it will definitely be very hard to obtain a FAPI that stays long enough in the tumor, but we are already going into the right direction, I can say. So, and then, so I told that FAP is only expressed on these cancer-associated fibroblasts, to say. But you also have activated fibroblasts, possibly in some wounds. what's the effect when someone also has, for instance, large wounds, for instance, ulcerative colitis, which is actually the colon then, which is very inflamed and which also might have some FAP.
Okay.
But we're also working on that.
You have to stay out of there.
Indeed. Yeah. So we still need a lot of research to figure out the potential side effects.
But potentially it's very interesting also for companies. And
Definitely. So we recently got funded by the FWO, a project.
Well
indeed.
Done.
And it's in our advisory board are a lot of companies actually, which will advise us, of course, but potentially also in the future take over our compounds.
And are they already knocking on your door?
Not yet, but we hope so in a few years.
All right. Well, we'll cross our fingers.
Thanks.
I've seen as well that you use llamas to produce antibodies.
Yeah.
What makes llamas a good choice to do so?
So we, as humans, all have antibodies, but llamas have very special antibodies, which are called nanobodies. And these are very, very, very small antibodies.
Okay.
So an antibody consists of multiple parts, and they only have the part that we also use to recognize a pathogen.
Because nanobodies are used very widely.
Yeah.
They're all produced in llamas.
Yeah, true.
Okay.
So only llamas have them, and also to be true, sharks. But a llama is more feasible to use than a shark, so that's why. And there is at the moment not really an antibody towards FAP. So, and we want to have it because we could use it too for our assays. So
interesting indeed. In your abstract, you mention a Dr. Beauvois. Who is this doctor, and what's the main scoop about your review together?
So when I started my PhD, we had the idea to write a review on FAP, but also on similar proteases, so similar proteins that also can convert other proteins, which have a role in cancer. So we only focused on these proteases, but then we wanted to describe to the rest of the world which current therapies exist towards these proteins, and then also which directions we could go, possibly. And that was the scope of our review, And it was nicely published in Cancer, so,
Very nicely, well done, indeed. Is there some advice you would like to give to new starting PhD researchers? Or more than one advice?
It's a roller coaster ride, definitely.
Because you're in your
I'm now finishing up my third year.
Okay, yeah.
So
you have had the hardest year. They always say at the end of the second year.
I can definitely say that. From now on, it should go up.
I'm sure.
But it's very nice. You have the opportunity to learn a lot. There are some setbacks, most probably. But you should enjoy the ride. You get to know a lot of people, and I feel more ready to go to industry. But because probably I will go to industry, I have the idea now. So
all right.
That's what I can say. Then definitely go for it. It's really nice. So.
Well, okay. Go for it, indeed. And is there something else you would like to share about your research now that we didn't ask?
So my main focus is FAP, and in our lab we do a lot of recombinant production, which means that we make our own proteins. We produce them ourselves because it's cheaper than commercially buy them. Okay. And also the quality is better, to be honest. So,
okay, I'm cheering a lot in this podcast.
It's often not from the first try, but eventually. And I also produced other proteins. Okay. For instance, TMPRSS2 is something totally different. It's present on extracellular vesicles in the semen. So I also managed to produce it. However, it was way more difficult than FAP. Okay. But eventually I did it. And it's also now under review for publication. So, yeah.
And so other researchers can come by and maybe ask for...
There is already a potential collaboration because I managed to produce it.
Okay. All right. And here internally in the Antwerp or...?
No, from the KU Leuven.
Okay.
Yeah. Yeah. But still, happy to have some interest in my production. So,
of course. It's doesn't matter where
And
the
I got
research
to know
is
the
done.
person on a conference in Italy.
Oh, well.
So
All right.
as a PhD student, you can also go to conferences
Yeah.
in very nice places.
That's where you
With
build a large
very
part
good
of your
wine,
network.
yeah.
Oh, very good wine.
Yeah.
And other things.
important
Tiramisu.
Yeah.
No. All right. Well, I think you can conclude. Thank you, Emile, for being here.
Thank you for the invitation.
Thank
It
you
was
very
a
much.
pleasure. Yeah.
Thank you and good luck with your presentation this afternoon.
Thank
You're
you.
the last one.
I'm the last one.
But
I'm
I'm sure all the students will be very interested after hearing your...
And the last one is the best one. So let's say that. Thank you.
Okay,
All right.
thank you.
Thank you.